Okay, I believe that we can start now and people can join afterwards as well. First of all, welcome all, and we are really happy to host you on fourth RefreSCAR Webinar Spotlight on series supported by RefreSCAR project. The title of today's event is Advancing Food Safety Management, Bridging Research, Innovative Technologies and Regulations. We are happy to support and present to you three speakers who will be sharing their own insights and expertise in food safety management. Before we begin with the event, I will just give you a quick overview of housekeeping rules. We want to notify all attendees that this session is being recorded because we want to make sure that people who are unable to attend can access this content afterwards. Please mute your microphone so we do not interrupt the presentations. Please close your camera during the presentation session. Also, throughout the presentations, you have chat available at your disposal should you wish to pose any questions during the session. And we encourage you to actively participate, especially during the Q&A session, where you can always raise your hand and pose any questions that you may have to our presenters. The agenda of the event is we will give you a brief introduction to our project RefreSCAR, which will be followed by 15-minute panel presentations. And the panel presentations will be followed by open floor discussion and closing remarks afterwards. To give you a quick introduction, RefreSCAR project is coordination and support action project funded by European Union, spanning over 4 years. It began in September 2023 and will last until August 2027. More details you may find on Cordis Europa website, but also our information is available on SCAR Europe site. The overall aim of the project is they strengthen SCAR within the European research area and to facilitate improved coordination of national and European bioeconomy research programs in the European research area through strengthened SCAR working groups, with the following specific objectives. One is to provide scientific and administrative support to the work of the SCAR working groups, to enhance inclusiveness within SCAR working groups and support active engagement of their members, to connect actors across research and innovation, policy and practice, with the aim of enabling SCAR to become a key bridging initiative across these domains, to help SCAR working groups to reach a broader audience of stakeholders and strengthen linkages with key EU and international initiatives, and to support the development of a strong SCAR foresight process, including the effective dissemination of its outcomes. And to this end, RefreSCAR is offering 6 pillars of activities. One is administrative support for meetings, workshops, and conferences, followed by scientific support, which involves portfolio analysis and studies. Also, we provide a wide range of communication support, which involves support for the websites, templates, reports, flyers, videos, and infographics. Also, RefreSCAR boasts with SCAR Engage program, which involves active engagement and action plans and supports for countries which are currently underrepresented in SCAR. Foresight process is the 5th pillar, which offers operational support and lessons learned, which are further to be disseminated across SCAR and wider. And the last but not the least is SCAR Connect, which involves bridging the gaps between research and innovation, policy and practice, stakeholder and network analysis, as well as organization of policy and practice events. And this webinar is one of the essential activities within our SCAR Connect support. RefreSCAR boasts with a wide range of consortium members across Europe, with the joint experience of over 100 years. And the members involve research performing organization, organizations, ministries, experts in innovation support, experts in project management, experts in communication, and also experts in capacity building. And together we are all working to achieving our RefreSCAR objectives. And I will get back to our webinar now. And I would like to give you a quick introduction to our speakers today. Firstly, I will start with Stephen J. Lehotay, who is a research chemist and lead scientist at the USDA Agricultural Research Service in the US. So Stephen is a lead scientist within the USDA Agricultural Research Service at the Eastern Regional Research Center in Windmore, Pennsylvania. Since 1992, he has conducted scientific investigations and method development, research involving improvement in the analysis of pests.
Says, okay, replace current share or just go ahead. Okay, you can see that. And
yes, we can see your screen, but it's not in full presenters mode. So
there we go.
And now it is. Yes, we can see it.
Thank you for the introduction and inviting me. And it's not my normal talk. This is I'm a researcher and generally don't work in the management side of things, but it's nice. I've given an overview of our programs before, and I'll go over it how it works in the USDA Agricultural Research Service. We are an arm of the USDA that does research, not regulations. And if I happen to mention any brand or firm name that in this talk, it does not constitute an endorsement by the USDA, but others of a similar nature. Contact me if you need to. I'll provide slides or other information if you just contact me. And I want to acknowledge my stakeholders. I've been in this field for 33 years at the USDA. Within the USDA, there's several branches, departments. So Food Safety and Inspection Service, Agricultural Marketing Service, Agricultural Plant Health Inspection Service, Foreign Agricultural Service. This is the National Institute of Food agency. I can't remember offhand, but they do funding, and this used to be. It's now part of AMS, and it's the Grains and Inspection, and ARS. So we have several within USDA, several within FDA in the US, several states. We have 50 states. I work with the states. The Pesticide Data Program has been a great program to work with over the years that does pesticide analysis and monitoring for risk assessment and reassessment of that's a nice program, actually, if you want to look into it. And IR4 is another one that those of you who are in the field should learn about if you don't already know. And then I work with producers, industry. That's all the companies involved in agriculture and in foods and reagents and instruments, and then international organizations. So it's basically anybody who eats food is somewhat of a stakeholder, and there's a lot of groups. So it's a big scope, and it's actually nice to be in this field. It's a niche, but it's also a broad niche in regards to how it affects people. So this is the structure of the U.S. government. Everybody in the U.S. should know this. We take civics classes, but I'm in the executive branch under the president. Here's the homepage for the USDA. We're one of several cabinet level positions. The Secretary of Agriculture reports to the president, and there's 13 others. And the Health and Human Services is where the FDA is, the Food and Drug Administration. And food is kind of separate from drug. And there is a push every year to combine agriculture and food together away from the drugs. But over the last 40 years, it hasn't happened. So the USDA provides leadership on food, agricultural, natural resources, which is also important in our kind of work. Everyone should be aware of climate issues, rural development, nutrition, and related issues based on public policy, the best available science, and effective management. And so within the USDA, we have several agencies, and within those agencies, food safety is one of the top priorities. And you can see the others. And I've highlighted the ones that relate to my field and kind of work I've worked with over the years, the different organizations. And here we are in the research, education, and economics with NIFA and Economic Research Service, National Agricultural Statistical Services, and the Agricultural Research Service. If you want to look into the US ARS more, there's our homepage, and we deliver scientific solutions to national and global agricultural challenges. That's our mission, and it's a really nice to be involved in such a mission. It's important and needed, and it gives a good feeling for me to work in this area in this agency. So food safety is one of the national programs, along with quality and nutrition. They've combined those aspects into, there's 4 national programs, one of which is here, and you can see the others. So Agricultural Research Service has national programs that kind of coordinate, and you'll see, I'll show you the national program approach. But within the national programs, independently, the scientists have an important role, and I look at this as my mission. We have to recognize real problems, address them, assess them, and strive to truly solve them. It's not just research for the sake of research or publications for the sake of publications. If we're successful, we transfer our work and implement solutions. And there have been scientists in my agency who have made revolutionary contributions to the agricultural revolution. And it's still happening. It's just not to the same. It's more esoteric now, I suppose you could say. But here's how it works to decide what our research should be. And the national program leaders, we have 2 in food safety, and they have this handbook that goes through the process. And here's the website if you're interested in getting this. It's a 20-some page report, I think. And you can get it here and download it. And this is figure 1 in the report. And OSCAR is the Office of Scientific Quality Review. And it starts anywhere. It's actually usually here. So we'll start here. And we get stakeholder input. And you saw the stakeholders. We have many stakeholders in my work. And they are invited to meet with the national program leaders, and scientists are invited oftentimes. We're just now starting our next 5-year cycle, and we had those meetings last summer. And they develop an action plan, and I'll show you that soon. And within that action plan, the scientists work with the national program leaders to devise their 5-year plan objectives. So they come up with what they want to accomplish in that. And then there's a review of a proposal. So we write a proposal and develop a project plan. And then they invite a review process. And if you are interested in being an external peer reviewer, you can fill out a form and submit it. And you can review the research plans if they invite you. And it would be based on your specialty. What's the specialty of the scientists involved? And then we conduct the research over 5 years, and every year we write a progress report. And then every 5 years, there's a review of what we accomplished. And if there is unsuccessful, we have this milestones and so on. And if we're unsuccessful, it hurts our performance, but also will potentially cause us to go into a different topic, or even lose our jobs if it doesn't work out so well, and that research area is no longer needed. So there's the cycle, and it goes through this, and we're currently in our next 5-year plan now. I have to write our project plan by May. And I have other scientists working with me on that. And Agricultural Research Service is all across the USA, and food safety is shown here. There's more than food safety at these locations, but we're spread out across the US. And here I am in Winmore, Pennsylvania. We have several working here on food safety at my center. No, there we go. And if it comes to chemical residues, I've highlighted Fargo and Winmore as the main players, and then Peoria does mycotoxins. So within food safety, the mission division is to enhance and protect public health in agriculture through the development of technology, strategies, and data that safeguard food from. So we work with not just me, but toxins, pathogens, chemical contaminants are the main issues, and that's very broad. And then during production, processing, and preparation, so it's anything to do with food supply and food safety. And there's also people working on nutrition and quality within not the national program, but within. There's even those working on soils and so on. So the 2025 program is covering 7 factors, including antimicrobial resistance that Kaye will talk about. We still have meetings and so on about it, but it's interesting that we have funding issues, and I'm in here, by the way, and I'm in number 4. And this is the next 5-year program. You can get this online. There's a whole report about it, and now I'm under technologies to detect, quantify, and characterize chemical and biological contaminants. And they noticed that antimicrobial resistance hasn't, we don't have the funding to continue, at least to the extent that was listed. But within these, antimicrobial resistance is still an issue that has to be addressed. So it's also consolidating. If you might remember, there's 7, now there's 5, but within the 7, there's the same. So this is our program of scientists and what their topics of research are. And it's microbial is the big issue. Since 1993, we had a real serious food problem that killed some children. It was the Jack in the Box scandal that led to the change of the program mainly from chemical residues over into microbial. And notice that also we're getting, over the years, computational and AI and those kinds of, that kind of research is gaining prominence. And so we're shifting from traditional biologists and microbiologists into more heavy data scientists. But we also have engineers, chemists, physicists, and so on, and including soil scientists, and depending on what their research is. And you can see here listed in terms of chemical residues. Here is my group. There's 3 of us, 3 SYs. We're called SYs. That's principal investigators, and we work on technology development, evaluation, and then there's other on chemical residues, chemical issues. Okay, so this is my group, and that's an overview, and my time is up. So I'll just note that I do the veterinary drugs and pesticides, and Going Chen in our group does metals and toxic metals, and emerging contaminants is Yelena Sapochnikova, and she's been very busy with PFAS lately, which is the hot topic. So I'll leave it at that.
Even you can continue. It's fine. You can continue until 30. So it's fine. Please go ahead.
I have a little more time. Okay.
Yes, please go ahead.
So yeah, this is our the support chemists and the scientists. So generally we have 1 support chemist per scientist in my agency, and or support microbiologist if they're working in that field. And then we have postdocs where we get funding for postdocs through internal programs or external. We can get grants and so on. NIFA is one of, but we can't directly go with USDA. We have to work with a land grant university usually to get funding external. But there's different funding sources for us as well. So chemical residues. I was invited in large part due to pesticides and chemical residues and pesticides, veterinary drugs, environmental contaminants. We also work on mycotoxins as in method development. And then there's anything else that is chemical residue related. Microbial is a different category, like pathogens and so on. And then there's emerging and metals, as I mentioned. And so with chemical residues, we keep the benefits in mind and how agriculture it's important to have good yields and good quality and so we shouldn't just regulate them and get them out. We just have to recognize the problems with chemical residues in food and the environment, and we have to have the means to manage, monitor, and mitigate. The triple M. I used to say some other words, but then I got a comment from a fellow in England who told me, oh, you should keep with the M theme and use these words. And I said, oh, that's a good idea. So we should do so, and it has to be within a reasonable cost. Food is perishable, and the methods we develop, we don't want to add to the cost or the time involved. So within my program, we develop and transfer to stakeholders effective, efficient, and useful analytical approaches for the screening, quantification, and or identification, which is just as important or more important than quantification, of chemicals of concern in food and food-related matrices. So anything to do with chemical residues, we want to develop methods that are easy and fast and effective and inexpensive. And so there's many reasons that we need to develop these methods. Compliance enforcement is number one. There's laws related to the tolerance levels in the US or maximum residue levels around the world. And all these laws were developed to protect the environment. Of course, there's international trade issues. I think there's 18% of the international trade relates to food. So it's a major reason that we need good analytical methods that are fast and easy and meet these needs. And then there's risk assessment for toxicologists, re-registration of pesticides and veterinary drugs, antimicrobial resistance I mentioned earlier. And then there's other aspects of residues with allergies and other toxicological effects. Interestingly, organic food, if you need methods to ensure that organic production is being done properly, or if there's no residues in a product that is marketed as such. And then there's, the instill confidence in food safety, and there's concern with food terrorism. And so I mentioned this in passing, but we want to, the best way to save money is to broaden analytical scope. So we develop methods that are mega methods to look for veterinary drugs, pesticides, and other contaminants in the same analysis. And by doing that, you combine methods in laboratories even in programs into a single place for the same sample. And that saves a great deal of money compared to older ways. And we want to meet detection limit needs, make sure of our identification and qualitative processes are good, and basically do a good job within, so I mentioned this, that analytical scope and data quality must be important. So in conclusion, the Agricultural Research Service serves to research agricultural problems, including food safety. Chemical food safety it needs highly efficient methods and operations, and that's where we come in. And big money can be saved by developing mega methods. And I use my mega method is catch reserve. catchers as a pesticide method, more or less, and now we're doing catchers, which is more than catchers. And then we have other research related to screening, which I didn't talk about or really mention much, but screening is also important. And I have specific projects that we're pursuing. So thank you for letting me go till 8:30, and it's about that time. So thank you.
Thank you so much, Stephen. If you feel urged to pose questions, please feel free to post them in the chat, and we will save the discussion for a Q&A session. And I would like to give a quick introduction to our next presenter, which is Kaye Burgess from Teagasc, and I will just shortly present her biography. So Dr. Kaye Burgess is a principal research officer in the Food Safety Department in Teagasc Food Research Centre, Ashtown in Dublin. Kaye's research group is focused on understanding the behavior and virulence of foodborne pathogens and antimicrobial resistant microorganisms in food productions using a One Health approach. She currently coordinates the Irish Department of Agriculture funded Port Assure and DETCON projects and was also a funded investigator as part of the One Health European Joint Programme. Kaye has a PhD in molecular microbiology from University College Cork in Ireland. I would like to ask Kaye now to start presenting her slides.
Thank you very much, Marina, and I'm glad you have recovered. So I'm hoping you can see my screen now.
Yes, yes, perfect.
Okay, so good afternoon, everyone, and thank you for the invitation to attend to present today. My presentation is focusing on the factors influencing antimicrobial resistance, or AMR, in agri-food production. This is a critical issue at the intersection of public health, food security, and environmental sustainability. And I'm speaking from a microbiologist perspective, working in microbial food safety and agri-food production, so how it impacts on microbial food safety. Over the next approximately 15 minutes, I'll explore some key drivers of AMR, the challenges we face, and potential solutions. So firstly, I think it's helpful to set out some definitions. So antimicrobials are medicines that are used to prevent or treat microbial infections in humans, animals, or plants. There are 4 different groupings of them, namely antibacterials or antibiotics used to treat bacterial infections, maybe like strep throat. Antifungals used to treat fungal infections, such as candida. Antivirals used to treat viral infections, such as COVID, for example, and antiparasitics, which are used to treat infections caused by parasites, such as threadworms, for example. I will be focusing on antibacterials or antibiotics today. There is no doubt that the identification of the first antibiotic almost 100 years ago was a game changer. And it has made a massive difference in terms of life expectancy. And these drugs remain critical in clinical and veterinary care. However, we do face a problem. Antimicrobial resistance has been identified, and it occurs when a microorganism changes or mutates so that they're no longer susceptible to an antimicrobial used to kill them. So how does this resistance develop? So in its simplest form, the antibiotics that are used, they kill the majority of the bacteria, but a subpopulation survives. And this subpopulation is now resistant to that drug. And then these multiply. And then if they go on to cause an infection again, this new population is more resistant to that drug. And the drug is essentially ineffective if we use it to treat an infection caused by that resistant strain. So, but how does that occur? Well, one of the biggest challenges is the genes that encode antimicrobial resistance are often encoded on mobile genetic elements. And this means they can transfer the DNA with these genetic elements between different strains and species. You also get other factors like, we'll say, increased activity of certain pumps in the membrane, or mutations so that the target for the drug is no longer present. All of these different mechanisms can contribute to antimicrobial resistance to different drugs. And there is no doubt that antimicrobial resistance is a global crisis. Around 10 years ago, the Jim O'Neill-led review on antimicrobial resistance estimated that AMR would kill 10 million people annually by 2050. This paper that I'm showing here is a recently published study where they looked at the global burden of bacterial AMR since 1990 and have projected to 2050. And it's estimated that it will cost bacterial AMR will cost 39 million deaths between 2025 and 2050. That's 3 every minute. And from the graphs, you can see here that there's a predicted increase in all regions of the world. In agricultural terms, AMR threatens food safety, it disrupts trade, and burdens producers with rising costs. In terms of the environment, AMR impacts ecosystems as antimicrobials can persist in the environment, causing increased challenges there. So specifically talking about antimicrobial resistance in agri-food production, just like to give a little bit of a background and some context. First of all, it's important to acknowledge that antimicrobial resistance is an ancient natural phenomenon, and it's driven by microorganisms means themselves to protect themselves and stay alive, protect themselves from outside threat. But it has now been exacerbated. As we learn more about the composition of microbial communities in diverse niches, we're increasingly seeing how diverse and complex AMR is. We now know that thousands of resistance genes have been identified in hundreds of different species with to a broad range of antimicrobial compounds. We also know that what we do influences the resistance genes that are present in a microbial population, and that complement of resistance genes is termed the resistome. So that's a term I will use again. The simplest example of how we can shape the AMR genes present is the misuse of a particular type of antibiotic, driving the presence of resistance genes to that antibiotic. They select out for it. We know that the types of resistance genes present will be influenced by the microbial community structure in any given niche. And we also know that the presence of mobile genetic elements contribute to shaping the resistome. So all of these factors play a role in shaping the resistance genes present in any given environment. We know that antimicrobials play various roles in agriculture, especially in livestock, aquaculture, and crop production. And antimicrobial resistance can spread through various interconnected pathways. And you can see some examples of the pathways here. We know that humans can acquire resistant bacteria through contaminated food or through direct contact with animals. The environment itself also serves as a reservoir and a route of transmission. Previously, we have tended to focus on individual sectors. But to truly understand the impact of the various intersectors, how they influence each other, we need to consider them together. To effectively address AMR, we need to take a One Health approach. This is a concept which considers the interlinkages between healthy people, healthy animals, and a healthy environment. Food is right at the center of that nexus. So agri-food production is a really important consideration when we're considering antimicrobial resistance. When we think of antimicrobial use in agriculture, we often first think of livestock production. And looking here, we can see how AMR may spread in this type of setting. In our group in Teagasc, we've been working quite hard to look at the the influence of different agricultural practices can have on AMR in a given setting. One example is the dissemination of antimicrobial resistance genes through slurry spreading or the spreading of manure from different species, and how the different species can impact on that. Or also looking at how regulatory changes, in this case regarding the use of high levels of zinc oxide, may impact on the resistance genes that are present in the pig intestine. So we're undertaking research to understand all of these different pathways. We possibly tend to think less about the role plants may play, but I think this figure from Chen et al.'s paper really demonstrates how plants also play a role in antimicrobial resistance gene transmission, and how the plant microbiome can actually act as an interface between human and environmental microbiomes, representing a crucial pathway for human exposure to AMR. Also, in terms of plant production, it's important to remember that there are parts of the world where antibiotics are applied to food-producing plants, providing an additional selective pressure for resistance development. Several factors can drive antimicrobial resistance in agri-food systems, and I've just briefly summarized them here. One of the first that we think of is the overuse and misuse of antibiotics, such as administering incorrect doses or using them preventatively in healthy animals. Certainly, I've used the term regulatory gaps. What I mean by that is many countries have very stringent policies on antimicrobial use in agri-food production. Others less so, or where enforcement is lacking, and therefore where you tend to get more inappropriate use of antibiotics. We then have the potential issue of environmental contamination, where antibiotics can often end up in the natural environment, either in water bodies and soil through runoff, and how that may impact as a selective pressure on microbial communities present. And then we have global trade, which has the potential to spread resistant pathogens via animals, food and feed right across the world. So all of these can play a role on influencing the resistome that is present in any production system. A couple of years back, the European Food Safety Authority published a scientific opinion on the role the environment plays in the emergence and spread of AMR through the food chain. And really, first of all, this study, this opinion, it demonstrated the detection of AMR pathogens of critical concern in food production. So we can really say, okay, there is a problem here. I've selected just one figure from this publication, which to me clearly shows how complex it is to understand AMR and its profile in food production. All of these factors can influence the load and types of resistance genes that may be present, including the amount and type of antimicrobials used, the possibility of co-selection, the microbes that are present, the mobile genetic elements, presence of biofilm, measures such as hygiene and sanitation, the type of farming, and factors such as climate change. And the list goes on. All of these can influence, and it demonstrates how complex it is to really quantify the relative role of each of these in influencing antimicrobial resistance. But a clear message is that there are many knowledge gaps where we don't have quantitative data at the moment, and which we really need to address. Looking at some other factors that can also influence the resistome, I mentioned things like selective pressures. These drive bacterial populations to evolve and may promote the dissemination of antimicrobial resistance genes through horizontal gene transfer. And that's the transmission of these mobile genetic animals. And that may happen in the human and animal gut, or even in the environment. We have seen that the use of certain biocidal agents can influence susceptibility to antimicrobial compounds in some bacterial species. We've seen that the presence of heavy metals in the environment or used in feed can also influence the resistance genes. And this can happen through a range of different mechanisms, including co-regulation, co-resistance, cross-resistance, or again, this presence of biofilm, which can protect bacteria from exposure to the antimicrobials. So you can see that the understanding the interplay between the various factors and also between the various sectors is crucial. So what is being done? Well, policy and regulation is central to our response. Internationally, countries have put in place action plans to tackle antimicrobial resistance. And quite often these are multi-sectoral plans, and they come with specific objectives and targets in different sectors, as well as follow-up to monitor progress. We need that these are living documents. They need to be followed up on. And this is just a small snapshot of the plans that are in place around the world. Just to give an example of the steps that are being taken. So I'm now going to look a little bit more closely at Ireland's National Action Plan. And in Ireland, we're actually on our second National Action Plan on targeting antimicrobial resistance. And it is a One Health approach with each of the relevant sectors. Within the agri sector, there is a very clear focus on the prudent use of antibiotics. And Stephen, in his presentation, had the 3 M's. In our case here, we have the 6 R's. And there we need the right veterinary diagnosis of the disease to ensure that the right antibiotic is used on the right animal at the right dose for the right amount in time, to ensure that the effectiveness in curing the animal and also minimizing AMR development. Very much moving away from blanket therapy. This is complemented by ensuring that antimicrobials are stored and disposed of properly to minimize environmental contamination. All of this is coupled with increased awareness on enhancing biosecurity and preventing disease entry and spread. Very much taking a prevention is better than cure approach, preventing disease coming in the first place. In this case, you then don't have to treat the animals. And this approach is underpinned by legislation to ensure that it is followed. More broadly, the National Action Plan for Antimicrobial Resistance has 5 strategic objectives. And these mirror those of the World Health Organization's Global Action Plan. Within it, there are specific key performance indicators within each objective. And these objectives improving awareness and knowledge, enhance surveillance of antimicrobial resistance and antibiotic use, reducing the spread of infection and disease, optimizing the use of antimicrobials in human and animal health, and promoting research and investment in new medicines, diagnostic tools, vaccines, and other interventions. Within Teagasc, we're playing a role in the delivery of this action plan, helping to increase awareness and knowledge for stakeholders in the agri-food sector, undertaking surveillance activities for antimicrobial resistance and the antibiotics themselves in various production systems, and supporting farmers to implement measures to reduce the spread of disease and optimize animal and plant health. We're also very active in terms of research on diagnostics and various interventions to ensure optimal health in our food and feed production systems. With that, I will conclude. I'd like to thank you all for your attention, and I welcome your questions in the discussion section. Thank you very much.
Thank you, Kaye, so much. This is a very important and pressing topic, and I believe we will have a lot to think about in the upcoming years, and hopefully a lot to let's say, align across countries, because, as you mentioned, the regulations are essential in this case. And I would like to give floor to our next speaker, Dr. Milica Pojić. And before that, I will just have a quick overview of her biography. So Dr. Milica Pojić is a principal research fellow in the area of food science and technology employed by the Institute of Food Technology at the University of Novi, Serbia. Her primary research interests relate to new food product development, especially in terms of novel and alternative food ingredients and methods and techniques for engineering food products and processes. She is a member of the scientific board of European Industrial Hemp Association, editorial board member of the journal Discover Food, Springer, and editorial board member of the journal Food and Feed Research from the Institute of Food Technology. Milica, welcome. The floor is yours, and we can see your slides. So your slides are now in full screen.
Yes, thank you. Thank you for inviting me today. Although we are close to each other, I would say that our collaboration is still unfolding, but hopefully we will increase our collaboration. I would also like to add that although it is not obvious from my biography, my master and PhD thesis is in the topic I'm presenting today. And I would like more to discuss today about one very unique analytical technique, near-infrared spectroscopy, and how it is applied in food processing in general. And I would like to include also the development of this technology and how it evolves from its application in quality assessment of raw materials and food to very new and emerging applications which is related to food safety. For the beginning, I would like just shortly to introduce what is this technique for those who are not familiar with the technique. It is technology which is completely undestructive, and it uses electromagnetic radiation from near-infrared spectra, and it is based on a measurement of absorption of the radiation by the sample or a measurement of the transmittance also by the sample, in taking into account what type of machinery is applied for the measurement. What is also very important to note is that this is a not direct technique, and it is not. It cannot be used without prior modeling of the whole machinery and equipment. So in order to use it for a specific target application, we need firstly to develop a model which is developed based on the application of a standard or reference methodology. This technique started developing in last century when first infrared spectrum has been recorded. But I would say that very intense development of this technique started in 1980s when actually very powerful computers managed to allowed to use different and powerful softwares for development of calibration models. So in 1990s, development of artificial neural network started to be applied in this domain. And those type of models actually allowed storing a huge number of data within one model. And it has been used mainly for determination of moisture, protein, and oil in cereals and oil seeds. And those models actually appeared very stable with high accuracy. And since leading producers of near-infrared equipment developed the system to gather the data and spectral data from the users, it appeared that these artificial neural network models contained in their model data and from all over the world, and it appeared that one model can be very reliably applicable in different parts of the world. However, in the last 15 years, it appeared that this infrared spectroscopy mainly developed in terms of different hardware and IT solutions rather than different in terms of different applications. But also it is worth mentioning that from determination of protein, moisture, and oil, it started, different scientists all over the world started developing different models, which are not only dedicated to a certain constituent present in the sample, but also to determination of different quality parameters. So what is, sorry, just one moment. What is needed to mention is that when the sample is exposed to infrared radiation, it happens that energy of chemical bonds with hydrogen changes, but since the food sample is of organic origin, which contains a huge amount of different chemical bonds with hydrogen, it appears that different organic compounds absorb this type of radiation at the same wavelength. So the resulting spectra is, I'm trying to, the resulting spectra is not specific, and it cannot be used as such. So for the determination of the relationship between the concentration of target compound and absorbed radiation energy is only possible if different spectral processing methodologies applied to smooth the spectra, also to find different spectral features from these not specific spectra, and to apply different regression methodology techniques to develop a model which will allow application of this methodology. So as I mentioned, the application of near-infrared spectroscopy can be divided into traditional, untraditional application, but also we are now facing emerging applications. So traditional application is related, as I said, to determination of moisture, protein, fat, fiber in different food matrices. It is, I would say, the easiest application, since all of those components are naturally present in the sample. Also, one of the traditional applications is determination of authenticity of olive oil and honey. And also a determination of ripeness, sugar content, and acidity of fruit. And regarding untraditional applications is determination of polyphenols, then determination of texture and rheological properties of wheat flour, for example. But also determination of different toxic compounds, since we are all aware that our food nowadays is compromised by different contaminants and toxicants. So a huge number of research papers are already published regarding determination of mycotoxin pesticides. But also a certain number of papers which I today will talk about is determination of processing-induced toxicants, which are sometimes present in our food products due to different thermal processing during the production of certain food products. It is worth mentioning that in the very recent scientific papers, there are some examples how this technique can be applied for determination, for example, of microplastics in food and feed. Regarding toxic compounds in food, we can talk about toxicants which are normally occurring in raw materials. We are faced due to very extreme weather conditions. We are facing the fact that more and more our raw materials contains different type of mycotoxins, but also due to very intense agricultural production, pesticides residues are there is a risk of having pesticide residues in the raw materials. This presentation will talk about mostly about processing-induced toxicants, about acrylamide and 5-HMF, both compounds which are both produced during thermal treatment during food production, and they are worth mentioning because there's evidence that both of them are carcinogenic and neurotoxic and also genotoxic compounds. 5-HMF is an organic compound which are formed during baking, frying, or drying during food processing. And it is produced through Maillard reaction and thermal dehydration of sugars. And commonly it is found in food products such as coffee, dried fruits, different baked goods, honey, and fruit jams. And its presence in different types of food is regulated by certain standards. But also determination of this compound is not widely recognized in ISO or EN standard, but there is an official AOAC methodology for determination of this compound. I here presented the terms of different types, different authors of who try to develop calibration models for determination of this compound. Matrix, which is mainly used for the development of calibration model, is mainly honey, because this compound is a kind of on the basis of this compound we can see if the honey is thermally treated, and also we can conclude about the freshness of the honey, but also roasted coffee and black garlic, which is nowadays a very unique and popular food commodity, has also been used as a matrix for the development of methodology. What can be seen here is that the sample set which was used for development of calibration models is not so huge. It is in the range of 40 to less than 200 samples, which is in comparison to artificial neural network with models which contains more than several thousands samples. It is very low number. But I would say that still these results have scientific significance, and although the having results on this parameter in commercial settings can be very useful, I would say that there is still a lot of room for improvement, and how these results can be of broader commercial significance. It still remains to discuss on this. What is interesting that accuracy of this model is very high. In terms of acrylamide, it is a chemical compound which is also formed during thermal food processing if the temperature rises above 120 degrees centigrade, and especially with low moisture food products. And it is present and can be found especially in fried and baked products, especially in potato chips and French fried potatoes, but also in baked goods and coffee. Regarding regulation, there is a EU directive on establishing mitigation measures and benchmark levels for the reduction of the presence of this compound in food. And we have a dedicated reference standard for determination of this compound in food. And this standard is related to application of liquid chromatography and mass spectrometry.
Milica, sorry, just to jump in for a second. You will have 5 more minutes until...
Okay, I'm finishing. This is almost the end. Similarly to determination of 5-HMF compound, it is the situation in acrylamide detection by near-infrared spectroscopy. It remains almost the same. A small amount of samples in the calibration set, but also I would say a very satisfying accuracy of developed model, but still without any broader commercial significance, only scientific significance. So why the need for this rapid technology is, I would say that there is a huge chemical hazards in traditional methodology, because the utilization of different toxic compounds is required. All of those traditional methods are very time consuming and require specialized equipment and personnel. And they're not able to simultaneously provide the results of multiple parameters of quality, which is very frequently needed. Therefore, I would say that near-infrared technology has a very high perspective, but there's still a lot of challenges. First of all, there is a huge complexity in calibration model development. Also, there is instrumental variability and matrix interference, but also different regulatory and standardization barriers. In order to discuss about the perspectives, we are facing the need for development of calibration models for emerging toxicants, but also currently different chemometric approaches for development of models are emerging, and devices are being more user-friendly and portable, and we also need to discuss about broader inclusion in food safety regulations. Thank you for your attention.
Thank you, Milica. Thank you so much for giving us insight on how the existing methods can actually be integrated into novel applications. We will start now our Q&A discussion. And to this end, I would like to pose a few questions to our speakers, but I will also use this opportunity to ask the audience if they wish to pose any questions. They may raise their hands or type their questions in the chat, and we will read them afterwards. So I would like to start with Stephen. Stephen, you have provided us with an amazing overview of how the process of research in food safety is going in America. That's something that we are not very familiar with. And based on your experience, we would like to ask you, how long does it usually take to identify a potential chemical residue that might be threatening food safety until it finally becomes standardized item in routine food safety analysis? We've seen that research takes around 5 years, but how long before it actually becomes implemented in safety practices?
It varies by what the issue is, and acrylamide is an example that Professor Pojić brought up. And in 2002, it was came up as an issue, and it took actually the solutions don't necessarily come up through detection, but through mitigation. And it is needed. The detection methods are needed first. Toxicologists are then involved in deciding in risk assessment whether it is something that needs to be regulated. And then it takes at least 5 years for something to become an actual regulated product. PFAS is going through that now. It was, oh gosh, 15 years ago, I remember the first talks about PFAS being an issue, and there were lawsuits involved in the United States, and that pushed things along in a different way. But it did eventually, and now the EPA in the United States is starting to regulate PFAS. So 15 years in a case like that. And sometimes it never becomes an issue. Sometimes something is detected, and the toxicologists will say that it's not worth regulating. So there's only it is so variable, and it depends on so many factors.
You have also mentioned cost benefits in this case. As you said, food is perishable, and we would not want to increase additionally the cost of food production by overburdening it with testing. So in these terms, do you believe that methods are also, selection of appropriate methods can also influence the adoption of testing in the process?
Absolutely. A good example is that prior to early 2000s, there were regulations in the books for nitrofurans in foods, in slaughtered animals, mainly chickens and shrimp and so on. And they didn't have methods. So they had regulations on the book, but nobody ever could find it, and it didn't matter. Then they developed a good method, and next thing you know, it's billions of dollars of food trade were affected by the method had been introduced to find levels above it wasn't an MRL necessarily, but it was banned. So they found it at super low levels. And so that's an example where the methods are very important. And even now, the methods are very important on the accuracy. They have to be useful for the purpose.
Thank you so much. Milica, I believe you wanted to pose a question as well.
Actually, I just want to comment on what was said about mitigation. I agree completely about it, and also this regulation EU, which I mentioned about, related to acrylamide. It's about majority of the document is on mitigation measures and how to select a suitable potato variety, and what the conditions for potato storage and transport should be in storage facilities, and also about the monitoring of reducing sugar content in the time of harvest of potato, but also about recipes and process design in order to decrease the risk on acrylamide formation during food processing. So mitigation is also we need to think about, not only measurement.
Thank you, Milica. Mitigation is something that we haven't reflected in these presentations, but of course, I believe that ultimately mitigation is how we want to drive our further impact by realizing how these traces, chemical traces that impact our health, our environment, our food systems. And ultimately, the goal is to create effective mitigation strategies. However, they are not always possible, especially because we see, let's say, global trade influencing these factors. So decisions made on one part of the globe, which we do not have any impact on, actually have lasting effects in some other places, which I believe, Kaye, that's something that I would like to discuss further on with you, because antimicrobial resistance is a global challenge. And you have mentioned that multiple factors are actually contributing to widespread antimicrobial resistance, or sharing actually the resistance genes across the continents, across sectors. And I would like to ask you, can you identify some agricultural practices which are contributing to this? And also, do you have any examples of global trade which are exacerbating this problem?
Okay, thanks very much. So I suppose in terms of agricultural practices that may be contributing, one of the ones that we have the greatest understanding of is the overuse or misuse of antibiotics within production. You know, that they're being not used in a prudent manner, and this will drive the development of resistance. So that's one of the things that has been very much tackled, and the individual sectors have made massive improvements on reducing antimicrobial usage, and monitoring systems are in place now as well, so that we know the amounts that are being used, why they're being used, and that they're being used appropriately. That said, if you take an antimicrobial, and again, like, it's very important to emphasize that they play a very important role in ensuring animal health and welfare as well. You know, so the prudent use is important. You know, it's not that we can take them away completely, and if you take a drug out of a system, that's not to say that the resistance genes will disappear immediately or over time. Again, that is something that we're trying to understand. In terms of your second question on the examples of global trade, we, on a daily basis, we all eat foods that arrive to us from all over the world. And as I mentioned, the regulations differ in different countries, and that is then going to impact on usage, and that can then in turn have an impact on resistance. It's still difficult. There's not a lot of quantitative data on, we'll say, on imported foods. In terms of the harmonized monitoring program for antimicrobial resistance in the EU, they have now added imported foods like broiler meat and cattle meat, for example, at border control posts into the monitoring in the past couple of years. So more information will become available. We have seen studies in the US as well, where they have looked at some multi-drug resistant foodborne infections, where they have indicated that it was likely from either from travel, food consumed while traveling, or from imported foods. But I think there's more to come in that space to understand that.
Thank you so much, Kaye. I have also one question posed in the chat, which is, have you seen legislation result in a change in methodology used for testing being less accurate? So the question is in relation to the methods being used, should they are insufficient in terms of accuracy? Have you seen a reversed, let's say, case of legislations being changed due to inadequate methods being used?
I guess this relates to my topic, and I would say that the tying of the methods to the legislation is a problem for technology development. And in the past, the legislation was more or less tied. And you can see this separation in their policy and oversight coming from veterinary drugs versus pesticides. And in the pesticide world, we use our tradition is to use multi-class, multi-residue monitoring that might make sacrifices for one or two analytes in order to get the whole in a fast and easy kind of an approach. Whereas traditionally the vet drug world, they have specific methods for specific drugs. And oftentimes they even dictate the method in the past. But those times have changed. And the vet drug world is going into multi-class, multi-residue now, because it's just as important to know what you're missing. And if you're not able to do the monitoring, then it's not worth doing at all. I mean, you're wasting all your efforts if you don't really have the scope. So I don't think the technology is so much better and the methods are so much better than in the past that I would say the results are more accurate now, even in the multi-residues. And you use a screening and then you can do a follow-up. So you can use a specific method if it's an enforcement or regulatory purpose, whereas the screening kind of finds out what's there. And then there's non-targeted. That's what's coming now, is non-targeted instead of targeted, to look for more of what you're missing. So I would say the opposite of less accurate. I would say better now than certainly in the past, due to both regulatory understanding of the technology and the improved technology.
Thank you, Stephen, so much. I believe this also opens up a perfect spot to pose a question to Milica. You have described a known method being used in a novel field, which is food safety. So the adoption of new analytical methods in safety assessment is usually a long and arduous process. Can you identify several research points that need to be addressed prior to the application of near-infrared spectroscopy in routine safety analysis?
Thank you. I would like to think, since now we are in the area of artificial intelligence, that this development will be faster than it used to be for development of artificial neural network models for protein and moisture. What is needed firstly is to standardize the equipment itself, because there are certain differences in terms of spectral range. So if you are developing a calibration model for a certain spectral range, this spectral range should be actually the model for a certain spectral range should be applicable for different types of instruments. And also this transferability of models between the instruments of different instrument producers is also very important. But still, those type of compounds, they are still not, the commodity is not paid according to this compound. And as soon as, for example, wheat was started to fade according to the protein content, it speeded up this process. So we need also some kind of impose of policymakers to define what is obligatory compounds to be checked in a certain type of products. And I would say that this would definitely speed up the commercialization of this. Not actually, this calibration model is a commodity itself for a company who is developing this. So I think that still there is a lot of improvement needs to be done, especially in terms of instrument itself, and to allow transferability to calibration models between the instruments of different producers. So we'll see.
Thank you, Milica, so much. I will just jump in with an unrelated topic, and that is we have reached the end, but I would like to invite you all to stay a couple of minutes longer because we have several more questions and comments. I hope everybody's on board with that. Okay, perfect. So we have a comment from our project coordinator, Orlani Konakor, and she says, thanks for the excellent presentations, great insights into the topic. Is it possible to estimate the contribution of human versus veterinary antimicrobial use in antimicrobial resistance? I suppose, Kaye, this is a question for you.
Okay, thanks very much. I think certainly attribution studies are very much what is needed to be able to do that. Data is critical for that. So again, it will depend on the country. You need to know what antimicrobials are being used and where they're being used and when they're being used. We know that in certain countries, there's greater use of antimicrobials in veterinary use than there is for human use. So that's something that needs to be considered. But I think it will be possible to put a quantifiable contribution on it, but at the moment we're at the stage of the gathering the data, and this is something that would be somewhat of a knowledge gap, and we also need to understand the various transmission routes where people pick up these drug resistant infections from, and how we can deal with those as well.
Can I add my experience with in the U.S. We for 10 years my agency did antimicrobial resistance as part of the action plan, and it's still going to happen. There's good programs for the veterinary side, but when this issue first came up about 12 years ago, I guess in the United States with a big push from the administration at the time. I went to meetings about it, and we put it in our 5-year plan and hired a scientist, but the main funding went to the medical. All the meetings is mainly about medical sources, and the veterinary side became secondary. The food and environment was a secondary consideration for the funding. So the stakeholders spoke, and the NIH pumped all the money into that area, and this area kind of, they got its funding, but it wasn't the main push.
I think that would be fair to say on our side as well.
Which is actually probably a missed or a blind spot overall, because while antimicrobials are extensively used in human medicine, they're actually used large scale in veterinary medicine. And also from my research experience, these actually can be the major source of antimicrobial resistance gene contamination. A perfect example was colistin use, I believe, that was being used extensively in China. And one of your colleagues actually was the advocate to banning the use of colistin in animal husbandry routine use. So not in prevention of the disease, but rather to make sure that they have a greater number of animals and the larger size of the animals, if I'm not mistaken. I will also read a comment from Jesus Escudero in Spain. He says many thanks for these high-class presentations and to share your ideas and insights into the topic. I would also like to thank you for these presentations. It's been amazing, and I believe we have opened several questions. Are there any further questions from the audience? Feel free to raise your hand, because this is the opportunity. If there are new questions, I would like to thank to all three presenters for taking the time, for preparing, and for joining us today. Thank you for sharing your insights, and I believe these events will provide insights into how we can actually better bridge research and how to promote research into efficient policies, and how we can actually globally interconnect as we are interconnected, as we can see. Effects from one country can spread easily and can be transmitted further across the globe. That's why I believe we need to work jointly. And also, when considering application of new methods, we also need to think about how efficient and how fast and how widespread can it be in the further utilization. Thank you all, and hopefully seeing you in upcoming webinars supported by our project RefreSCAR. Thank you.
Thank you for,
Thank
I
you.
enjoyed it.
Thank you.
See you soon.
Thanks.